Hello, all. My name is Dr. Partha Biswas. I'm an associate professor at the Department of Medicine, University of Pittsburgh. I'm also a secondary faculty member of Department of Immunology and a graduate faculty member of program in Microbiology and Immunology. My lab is located at BST South Tower at University of Pittsburgh. The research focus of my lab centers around understanding immune response in the kidney. And that's why we call ourselves renal immunologists. As you know, kidney disease is a major problem in the United States. Around 15% of US adult population in 2017 suffered from some form of acute or chronic kidney diseases. And the number is going to rise due to increased prevalence of blood pressure associated problems and diabetes. So over the decade, understanding immune response in the kidney has always lagged behind. And this is primarily because kidney is an organ which is where studying immune response is a challenge. This is primarily because of poor regenerative capacity of the neurons, blood pressures, fluctuations, as well as presence of uremic toxins, all of which confound the immune response in the kidney. We took advantage of this position and placed ourselves in the renal immunological world to understand the immune response and the consequences of immune response in the kidney. Currently, there are four major projects in the lab. In the first project, we are studying the role of inflammation and particularly inflammatory cytokines in the pathogenesis of autoimmune glomerulonephritis, which is a clinical manifestation of many autoimmune kidney diseases, including lupus nephritis, ANCA vasculitis, and Gutt-Pasteur disease. We are primarily interested in studying the inflammatory cytokine interleukin 17 in the pathogenesis of autoimmune glomerulonephritis. We showed that mice deficient in IL-17 receptor signaling are protected from autoimmune glomerulonephritis, indicating a pathogenic role of this inflammatory cytokine in the pathogenesis of autoimmune glomerulonephritis. Subsequently, we were able to show that neutralization of IL-17 can be an important therapeutic pathway to treat autoimmune glomerulonephritis in these animals. Although IL-17 has always been linked to many autoimmune diseases, it has some beneficial role because it protects hosts from fungal and bacterial infection. And kidney is an organ is also susceptible to many bacterial and fungal infections, including Candida albicans, methicillin-resistant Staph aureus, and we showed that IL-17 plays a beneficial role by protecting the kidney tissues from destruction following these infections. In the third project, we were interested to understand the role of IL-17 in renal fibrosis, which is an inevitable outcome in many chronic kidney diseases that results in death and loss of renal function. We showed that IL-17, by activating kallikrein pathways, protects against renal fibrosis for the first time. And finally, we were currently interested in understanding why patients with kidney diseases are susceptible to many infections. In fact, 20% of the mortality in patients with kidney disease and undergoing dialysis are due to infections. So using a mouse model of kidney disease and associated uremia, as well as patients undergoing dialysis, we showed that uremic toxins, which accumulate in the blood due to improper kidney function, have a negative impact on the function of neutrophils. Now, these neutrophils are the primary line of defense, and it plays important role in the clearance of fungal infections. Now, we showed that the neutrophils are unable to clear the fungus due to a defect in the metabolic pathways, that results in increased susceptibility of mice against invasive fungal infections following Candida albicans infection. So these are the 4 projects that we are currently interested in lab, and lab rotations will be available in 2021. And so if you are interested to know about our research, please follow us at Biswas Lab at Pitt in Twitter handle, or email me at psb13@pitt.edu. I wish you all the best, and I hope you will like Pittsburgh. Bye.